Hi everyone. In the previous part, we had gone through the one of the type of phosphorylation, that is substrate level phosphorylation. Now, in this part, we will discuss in detail about oxidative phosphorylation. Okay, so let's have a quick review of what is meant by phosphorylation and what are the types of phosphorylation. As we know that phosphorylation is a biochemical process that involves the addition of phosphate to an organic compound is known as phosphorylation. That means addition of a phosphate to an organic compound is going to be called as phosphorylation. And this biochemical process is going to be carried out through the action of certain enzymes called as kinases. That's how we have seen in the substrate level phosphorylation as pyruvate kinases, isn't it? Or phosphorotransferases. And we know this phosphorylation is very important in certain reactions or the chemical process like cell cycle, cell growth, apoptosis, and signal transduction pathways. So these three modes of phosphorylation that we have discussed is photophosphorylation and substrate level phosphorylation in the previous part. Now we are going to discuss in detail about the oxidative phosphorylation. So what is meant by oxidative phosphorylation? It's a mechanism that occurs in aerobic respiration obviously, and in which ATP is synthesized at the expense of proton motive force. What is this proton motive force? We will discuss now. So what is a oxidative phosphorylation? It is a process in which ATP is formed as a result of transfer of electrons from NADH and FADH. So these are going to be of electron donors which are going to give the electrons to the oxygen through a series of electron carriers is going to be called as oxidative phosphorylation. Or this can also be defined as production of ATP at the expense of proton motive force by electron transport system is called as oxidative phosphorylation. Or in a simple terms, synthesis of ATP in the presence of oxygen is going to be called as oxidative phosphorylation. I think so you are going to opt this definition because it's very easy to remember. The synthesis of ATP in the presence of oxygen is going to be called as oxidative phosphorylation. So we can have the definition in any of the three types, which is one and the same. In one term, we are going to tell that electrons are going to be the donors from NADH or FADH, and we are also going in some other, the proton motive force is involved in the synthesis of ATP, so that is also called as oxidative. And in another process, we are saying that the synthesis of ATP is going to be in the presence of oxygen, so that is also going to be called as oxidative phosphorylation. Inorganic phosphate take the energy released by the oxidation of substrate and is converted into energy rich phosphate, that is nothing but the ATP. So this phosphate is transferred to ADP, and ATP is formed. And the oxidative phosphorylation generally occurs in the plasma membrane of prokaryotes. But coming to the eukaryotes, this oxidative phosphorylation is going to occur in the mitochondrial membrane. So whatever the thing, it may be a prokaryote or eukaryote, it involves the enzyme called as ATP synthase, or it is also called as F0 F1 ATPase. Okay, this is a special enzyme that is involved in the oxidative phosphorylation. Why this oxidative phosphorylation occurs only in the plasma membrane or the mitochondrial membrane of eukaryote means these enzymes that are essential for this oxidative phosphorylation are going to be located in those areas. So obviously that this oxidative phosphorylation occurs in those regions only. Okay, so let's see the structure of this ATPase, how it is going to function. So this is the structure of ATPase or ATP synthase or F0, F1 ATPase. Anything is the one and the same. The proton motive force that is derived ATP synthesis involves a catalyst, which is a large membrane enzyme complex called as ATP synthase or ATPase. So what is meant by proton motive force? Just have a glance of it and then we'll move back to the structure of it. Okay, look at this picture. When oxygen is reduced to H2O after accepting the electrons transferred from electron transport chain, it requires protons from the cytoplasm to complete the reaction, isn't it? These protons that are going to be originate from the dissociation of water molecule, that is H2O into H plus and OH minus ions. The use of these protons in the reduction of oxygen to water and the extrusion of this throwing out of protons outside the membrane during electron transport chain causes a net accumulation of OH minus ions inside the membrane. Despite of their small size, because they are charged, that means positive charge and OH minus is having a negative side charge, inside the membrane there are going to be of neither H plus nor OH minus freely passes through the membrane. So equilibrium cannot be spontaneously restored on both sides of the membrane. So this non-equilibrium state of protons and OH minus ions on opposite sides of the membranes results in the generation of a pH gradient and an electrochemical potential across the membrane. With the inside of the membrane, there will be more negative charge because of OH minus, and outside of the membrane, we are having the more positive because of the more protons. That means positive and acidic. This pH gradient and electrochemical potential cause the membrane to be energized, and this state of membrane, which is referred to as proton motive force, that is PMF, proton motive force. And generally it is going to directly drive the formation of ATP ion transport, and somehow it is also involved in flagellary rotation and other useful works also. Now this proton motive force is very essential for the synthesis of ATP in the process of oxidative phosphorylation. And this proton motive force, everything is going to be occurring, and the ATP is going to be synthesized by the special type of the enzyme, membrane embedded enzyme called as ATPase. So let's see the ATPase structure. Okay, and as I said, the proton motive force, that is derived ATP synthesis, involves a catalyst. So here it is the catalyst, which is a large membrane enzyme complex called as ATPase or ATP synthase. Now, this ATP synthase or ATPase consists of mainly two parts. How many parts? Two parts. One is going to be called as F1, which is located on the internal side of the mitochondrial matrix. Okay, and this is a multi-subunit headpiece. So we can consider this as a headpiece. That is, F1 is a headpiece, and it is going to be of a multi-subunit. And this is going to be present in the mitochondrial matrix coming to our eukaryotes, but it is going to present on the cytoplasmic side in the prokaryotes. And then coming to the second part, that is F0, a proton conducting channel called as F0, that resides in the inner membrane of mitochondria, especially in the eukaryotes, and the same thing, it is going to be in the plasma membrane in the prokaryotes. Okay, now the ATP synthesis takes place at the F1 F0 ATP, which is the smallest non-biological motor. So in the living organism, the smallest non-biological motor is going to be this ATPase or ATP synthase enzyme. Now the F1 is the, as I told you, is a multi-subunit headpiece, and it is catalytical complex responsible for conversion of ADP into the ATP. So here is our head part, which is going to be involved in the conversion of ADP into ATP. This is not simple as I am saying. Okay. Let's see what are the components of this F1 headpiece part and what are the components of F0, that is the tail or the stalk of this part. It looks like a lollipop if you're in the inverted manner. Okay, so let's see in the F1 part. As I told you, it's a multi-subunit headpiece and it is a catalytic complex, isn't it? And it contains of 5 polypeptides, 5 different polypeptides. What are those? One is alpha, which are 3 in number. So alpha 3. Then we had second one is a beta. They are also 3 in number. Then we are going to have a gamma 1, epsilon 1 and delta 1. So that's how many polypeptides are there? 5 polypeptide units are there. One is alpha, beta, gamma, epsilon and delta. Alpha are 3 in number, beta are 3 in number, and you are going to have epsilon 1, gamma 1 and the delta 1. Then coming to our next part, that is F naught. F naught, we know that it is integrated in the membrane. So this is our plasma membrane or the mitochondrial membrane that is embedded in it. And it consists of 3 polypeptides. F naught contains 5 polypeptides. Sorry, F1 contains 5 polypeptides, alpha, beta, gamma, epsilon and delta. And the F naught contains 3 polypeptides. So what are those 3? A, B is going to be the others. Here it is a B and here is a C. A will be 1 in number, B or 2 in number. Coming to the 3, that is C, it is of 12 in number. So that means the C subunits are going to be of 12 in number. So this is all about the structure of ATPase. So it consists of 2 parts. One is F1 and another one is the F0. F1 is the head part, which is a multi-complex catalytic part containing of 5 polypeptides, whereas the F naught is the intermembrane, that is integrated in the membrane, either the plasma membrane or the mitochondrial membrane, and consists of 3 polypeptides, that is A, B and C. A is only 1, C is 12 in number and B are 2 in number. Okay, so this is all about the structure of an ATP synthase or ATPase. So let's see what is the functioning of this ATPase, how it is able to convert the ADP into ATP. Okay, let's see. For example, in E. coli, the subunit A, where it is, here it is going to be. The subunit A is responsible for channeling the proteins that are present outside the cell. The protons are going to be channelized into the cell, okay, through this one. So it is going to channelize the protons across the membrane. So now during this crossing, the subunit B protrudes outside the membrane and forms along with B2, and you are going to have the delta subunit as a starter. So these are responsible of starting. So here is the delta and here is the B and here is the A. So when the protons are entering, so it is going to push the B subunits along with the delta subunit, and these 3 together is going to start up the starter. Now what is happening? Proton movement through A subunit of F naught drive rotation of the C. As soon as the protons are moving inside through the A, it is going to exert some sort of a rotation in these C particles, which are 12 in number. So there will be a rotation. And this rotation is going to generate a sort of pressure called as torque, and that is transmitted to F1. So here is the F naught where the C is present. Because of the movement of protons, this C is going to have some sort of generating a torque, that is pressure, which will transmit that torque to rotate the internal part, that is F1, where you are going to have your gamma and epsilon part. So these are also getting rotated. Now as a result, so what is happening? Energy is transmitted to F1 through coupled rotation of gamma subunits, causing conformational changes in the beta subunit. So we know that we are having the beta subunits. So here there will be some sort of conformational changes. Once the conformational changes, what is happening in the B subunits allow for binding of ADP and inorganic phosphate. That means once the beta subunits are getting conformational change, and this conformational change will allow the binding of ADP plus inorganic phosphate. We know once these two combine, obviously we will get the ATP. So that's how we are going to have the conversion of ADP and inorganic phosphate to ATP. When this is going to be finished off, again the B subunits returns to their general or original configuration, and the rest of the things is going to be stopped. And in the same manner, again the protons are coming inside through the A subunit, and now this A subunit is going to transfer that to the B subunit and the delta, which causes some sort of a starter one, which creates some sort of a targ in the C2, which due to this targ it is going to mobilize, which is transmitted to the neck inside the what we call it as gamma and epsilon parts, where they are going to have some sort of a rotation, and this rotation is going to change the conformational changes in the beta subunit. If there are going to be the conformational changes in the beta subunit, obviously that will allow the binding of ADP and inorganic phosphate, which are converted into the ATP. So again, once the ATP is formed, the beta subunit comes to its original conformation. So this is how the proton motive force is involved in the synthesis of our ATP. I think so you understood very well. Okay, let's have a quick description of the structure, then you'll come to know the synthesis. So how many parts are there in the ATPase? Two parts are there, F1 and F0. In the F1, where it is the head part, which is of multi subunit catalytic complex, okay, and responsible for the conversion of ADP into ATP. And now this is going to have the 5 polypeptides: alpha, beta, gamma, epsilon and delta. Alpha are 3 in number, beta are 3 in number, gamma, epsilon and delta. Then coming to the F naught, which is integrated in the membrane and consists of 3 polypeptides, that is A, B and C. A is only 1 in number, B are 2 subunits and C is 12 in number. Okay, now what is happening when A is responsible for the channeling of protons across the membrane, while subunit B protrudes outside the membrane and forms along with B2, that is what we call it as a delta subunit, as a starter. Proton movement through A subunit of F naught, this is F naught, isn't it, which drives the rotation of C protons, generating a torque. So here you can see a torque that is transmitted to the F1 part. So here is our head part, F1 part, through coupled rotation of gamma and epsilon, which is present here. Now these subunits causing conformation changes, these subunits, that is gamma and epsilon, will cause a conformational changes in the beta subunits, and the conformational changes in the beta subunits leads to the conversion binding of ADP and inorganic phosphate, which is giving rise to ATP. And once the ATP is formed, the beta subunits return to their original conformation or configuration. So this is all about the proton motive force, how it is going to be utilized in the formation of ATP with the help of an enzyme called as ATPase. Okay, so a proton motive force and ATP synthesis. So this is what I have explained in detail. So you can go through all these points that we discussed. And the next point is ATP yield from oxidation of glucose in aerobic respiration. So in the previous part of aerobic respiration, I had told you that there are going to be synthesized of 36 or 38 ATP are being produced, but that is going to be only the theoretical what we call it theoretical value of giving the estimation of complete oxidation of the glucose molecule. But experimentally, it was proved that we are going to have the ATP of generation only 32 or 30 ATP from the oxidation of one glucose molecule. So what is the variation between the theoretical and the experimental? In the theoretical, the PO ratio, what is meant by this PO? The number of ATPs formed per oxygen is going to be called as PO ratio. This PO ratio is going to be of about 3 for NADH and 2 for FADH2. This is based on the theoretical hypothesis. But coming to the experimental research or the experimental value, here you are going to have the PO ratio, that is number of ATPs formed per oxygen, is going to be of 2.5 for NADH and 1.5 for FADH2. So that's how the ATP synthesized when glucose is oxidized to carbon dioxide is about 30, that is 30 or 32. Okay, so this is all you have to remember, that why the variations are going to be of 36, 38 or 30 or 32. But now it was widely accepted that one complete oxidation of glucose molecule is going to yield the 32 ATP molecules. Okay, and this ATP yield in bacteria, especially in the aerobic condition, can even be less because the electron transport system often possesses lower PO ratio than the eukaryotic system. This we have discussed depending upon the eukaryotic system, but in the bacterial system it may be even less because of the having lower PO ratios than the eukaryotic systems. Okay, so this is all how we can conclude that we are going to have the 32 ATP for one complete oxidation of a glucose molecule through the aerobic respiration. So with this we have finished the aerobic respiration concept of having the three stages, that is glycolysis, where the glycolysis of again of three modes, one is EMP pathway, ED pathway and HMP pathway, and then we had gone with the TCA cycle, then the electron transport chain, and then we had gone a very important topic called as phosphorylation, that is substrate phosphorylation and the oxidative phosphorylation. With this, we have finished all the concepts of aerobic respiration. Okay, thank you.